Dr. Marc Slattery. Marc got his bachelor's in biology at Loyola Marymount University, and then he got his master's in marine science at San Jose State University and Moss Landing Marine Laboratories. So yeah, he was a student here. He was in the invertebrate zoology lab of Jim Nybakken. I just grabbed his thesis from the library. It's entitled Settlement and Metamorphosis of Red Abalone Larvae. And while at MLML, he also participated in one of the Vertex cruises led by John Martin that was testing the iron hypothesis. So really been a big part of MLML's history. After he graduated, he had several professional positions, all related to marine science, research, or scuba diving. And then he pursued a PhD in biology at the University of Alabama at Birmingham. And he was in the lab of James McClintock and studied chemical defenses and population biology of Antarctic soft corals. So, yeah, lucky you, diving in Antarctica. Then he did a postdoc at the University of Guam before moving to a faculty position at the University of Mississippi, Ole Miss, where he's the professor of, I actually don't know how to say this word, pharmacognosy?
Yeah.
Okay, pharmacognosy and biomolecular science. And also is the director of the National Institute for Undersea Science and Technology. He's received many awards and honors in addition to his science. They're also related to his diving, so he does a lot as a dive safety officer, and also has done a number of technical dives that is that you in that photo?
It's actually my collaborator. But.
Okay, yeah. So please join me in welcoming Dr. Slattery as he gives his talk on the functional ecology of mesophytic sponges.
All right. Thank you for that introduction. Thank you for the invitation. I'm happy to be here. Before I get started, I actually want to apologize. I've had this on my books for 2 months, and I, of course, waited until about 2 weeks ago to write this talk, and we had an ice storm in Oxford, and it knocked out all the power for 2 weeks, and so I didn't get any of this done until I was on the plane on my way out here. So it's going to be a little rough, but we'll get through it, and yeah. Anyway, today I want to talk about deep reef sponges because I really love them, and as you can see, they're really, really impressive. My collaborator here is about 6'3", 250 pounds, and he is dwarfed by that zesto. And the question I get, even though I think I'm doing really interesting science, the number one question I get at meetings is like, oh, how do you become a deep diver? And so I'm going to talk about that very briefly because it gives me a chance to go down memory lane. Oops, I don't know how to advance. I'm not. I can thank my mentors here at Moss Landing from way back when. As Amanda was saying, I did a thesis 40 years ago on larval settling queues in red abalone. And from there, I went to UAB where I did my dissertation, as she said, underneath the ice, looking at soft corals and sponges, chemical defenses. That set me up for my postdoc in Guam, where I looked at chemical ecology of soft corals in the Indo-Pacific region. And now I sort of dabble in everything at Ole Miss, mostly in the Caribbean because that's close to home, but I get a lot of different places. And so the through on all of this is that I've done a lot of diving over the years in a lot of different systems. And I really love diving, and I think that it's the best way to do science. I really am kind of not impressed with what the vehicles bring back and stuff. But as I was reading about scientific diving, I sort of came to realize that most of the work is at 100 feet or less, and in many cases much less than 100 feet. And I'm the one who always kind of stuck his head over the edge of the reef and looked down, and the reefs don't stop at 100 feet. They keep going, and there's a lot of stuff going on down there, and I could see there was a lot of stuff going down there, and I really wanted to be down there and understand what was happening and how it fit into the larger subject matter. And so I started looking at how it was done, and back in the day, it was like somebody got hold of a submersible and on a research vessel that cost a lot of money, and then they go out and maybe they get a few pictures and maybe they get a few samples with a manipulator arm or whatever. Nowadays, I think they also have AUVs and ROVs that are doing the same thing. But the bottom line is, this is really expensive research, right? And most of us are not going to get that, particularly for the depths that we're talking about here. So I started looking at other ways. And initially I was kind of like, well, diving to 300 feet, that could be dangerous, and my wife wasn't too excited about it either. But I already sort of had a technical diving background. And that was, I did ice diving in Antarctica. For 10 years, I did cave diving in the Bahamas because I was really interested in sponges on the walls of the caves. I did a stint in Aquarius as a saturation diver. And all of those have one thing in common. They have either a literal or a figurative ceiling over your head. You can't make a direct descent to the surface. Okay? If you do, bad things happen. And when I started thinking about it, if we do deep diving, it's going to be decompression. It's the same story. So I said, okay, well, maybe we can pull this off. So in 2000, my collaborator, Michael Lesser, and I decided we're going to start doing deep reef research. And we decided to train up, be good. And you can see back in the day what we were using. These are twin steel 120s. Those are on your back. And by the way, you need all of those bailout bottles on your sides in case shit hits the fan and you got to get back to the surface. And both of us were in our 40s at the time, and it was like, this weighs a lot. We're not going to be doing this long unless we figure out a better way. And so we started looking into rebreathers. And there's a rebreather on my back. It's sort of about the same size as a set of doubles, but it's about a third of the weight. You still need a bailout bottle, but we had by then worked with our dive team to figure out ways to sort of distribute the load and be more efficient. And so we weren't carrying as many bottles down to depth. And there's a cost benefit here as well, in that the gases that you're breathing, when it's open circuit, you're losing about half of it as bubbles, right? But in a rebreather, you keep that going. And since helium and stuff is really expensive, that was saving us some money to do research as well. So about 20 years ago, we got trained over onto rebreathers, and we're really comfortable with them, and decided, yeah, this can work. And we haven't really looked back. Hundreds of dives now. And so now I want to stop talking about that, and let's talk about science. And I want to set up sort of the background context for what it is I'm going to talk about. So as long as I've been a professional scientist, coral reefs have been in a world of hurt. And there's dozens, if not hundreds, of papers that deal with this issue of phase shifts. And basically, this is saying that you're going from a state where it's coral-dominated to usually algal-dominated. But as this particular review talked about, there can be other things. And depending where you are, you can go from coral dominance to soft coral dominance, or coral dominance to corallomorphs, or, for the purpose of this talk, corals to sponges. Okay? And so we're going to be looking sort of at this issue of phase shifts. And in fact, about a little bit after that, Jamie Bell's group came up with interesting opinions saying coral reefs are going to become sponge reefs in climate change. Michael and I did a little more work on that, and I don't think it's going to happen because of the oceanographic conditions. We can talk about that later. But what I want to do instead is talk about this paper by Peter Glenn, which is really important towards the deep reef stuff. And he was studying coral bleaching in Panama over a depth gradient. And when I talk about a depth gradient, it wasn't big. It was like 20 meters or something. But what he was seeing was that the deep population was not getting bleached and the shallow population was. And it led him in this paper to ultimately suggest that maybe the stressors won't make it down as deep. Maybe the water is acting as a buffer. And the deep reefs will be sort of protective. They'll be a refuge. And ultimately, if everything goes well, they might even reseed the shallow reefs as they become degraded. And this became known as the deep reef refuge hypothesis, and it has huge implications for the field of research that I'm in. NOAA, they are the managers of aquatic resources. Somebody finally found this paper and paid attention, and they contacted us, a group of us who were already doing deep reef research, and they said, we're going to hold a workshop. We want to ask the question, are deep reefs potentially a refuge? And even more generically, how do deep reefs fit into the broader coral reefscape? Okay? And those are great questions. At this workshop, we came up with the term mesophytic reefs, and we'll talk a little bit more about that in a minute. From our perspective, there are really two important characteristics that we have to bear in mind. It's not whether or not the deep reef is a refuge, it's whether or not it's going to reseed the shallow reefs. So there has to be some degree of biological connectivity between the deep and the shallow reefs, right? And so larvae have to be dispersed to the shallow reefs. And that's something that we looked at and we'll talk about in a few minutes. The other issue is this issue of stability. Now, Peter Glynn noted that there was not as much bleaching in his particular system, but there are a lot of deep reefs everywhere, and his stability is common, because if they're not stable systems, then they're really not going to be able to, they're not a refuge, and they're not going to be able to do what we hope they would do. And so we'll talk about these things going forward. The only other thing I want to mention at this time, since this workshop about 15 years ago, we now have roughly 100 publications per year. So there's a pretty good positive trajectory of this research science. And that makes me really happy, because I think mesophytic reefs are really cool and everybody should know something about them. And unfortunately, NOAA has not really put the money into them in terms of getting us the research opportunities, and that's sad. But nonetheless, mesophytic reefs are awesome, so let's talk about them. So what are the mesophytic reefs? At the workshop, we decided they were anywhere from about 30 meters down to 150 plus meters. And I need to set this up by saying it was the NOAA guys who said it's going to be 30 meters. Okay? And their rationale for that was most of the research to date ends at about 100 feet. We don't want to be in a situation where you scientists tell us that mesophytic reefs start at 150 feet, and then we had another black box from 100 to 150 where nobody knows anything about it. So if we're going to end up funding this thing, we're going to be able to fund it from 100 feet on down. Okay? And so I understand why they did that. That doesn't necessarily make the animals at 30 meters mesophytic, but pragmatically, we get there. Okay, the 150 meters was more of us as scientists. Most of us, because this is an issue of light limitation, most of us had sort of bought into the idea that somewhere around the compensation point, 1% light level, is going to be an important characteristic. And maybe that's where mesophytic reefs sort of start dropping out. Okay? Turns out it's not entirely true, but at the time, that's what we were working with. We also knew that the 150 plus is because, depending on where you are in the world, it can be shallower for the 1% light level, or it can be much deeper. And so we just sort of set it up like that. Again, because this is light limited, we're moving from autotrophic systems, so plants and such, and corals, to heterotrophic systems. Okay? Again, intuitively, these deep reefs are further away from the stressors, so maybe they are better protected. We don't know that yet, but that's something that's worth studying. This issue of connectivity, we already know there's physical connectivity, right? There's internal waves, there's upwelling, there's stuff, there's an elevator that can bring propagules from the deep reef to the shallow reef. The question is the biological connectivity, and that's still to be resolved. And then you get to this deep reef refuge hypothesis and whether deep reefs are a seed bank. That's, again, to be determined based on different research teams. One other point, and this relates to the Caribbean, where arguably most of the mesophytic work to date has occurred, is that they are sponge-dominated communities. Okay, there's a lot of sponges down there. Highest diversity between 60 and 100 meters, and a lot of unique species. By our own counts, about 50% of what we see on the deep reefs are new to science. Okay? And we also see some interesting adaptations as well. And one of the things I'm interested in is over gradients, ecophysiological changes. So it's a very cool system to work in. Okay, so back to this issue, mesophytic, as the name implies, these are light-structured communities. Okay? And here you can see, again, those 1% light levels for, these are several published results around the world. They're not all of them by any means. But again, you can see low light situation here in Belize is 58 meters. The 1% level in the Marshall Islands is 102. So, there's a range there that we're dealing with. And that's going to vary from site to site based on optical properties of the water column. The other issue is the angle of the sun, the albedo, okay? And it's going to vary depending on reef slope. In this case, we have a vertical wall here, and an animal that's here is going to see less light than if he was sitting on some kind of a sloping surface. So light is impacted in a couple of different ways here. Okay, the other issue here is we've got this light gradient from shallow to mesophotic reefs, and so we're obviously going to see a shift from the autotrophic to the heterotrophic processes, which means we're also going to see a shift in biodiversity from the autotrophic sort of algae and corals to the heterotrophic, in this case, sponges. Okay, interestingly enough, we discovered there is a zonation pattern in the mesophotic reefs. And I'm not going to go into this in great detail. It's in this paper. But the bottom line is we looked at all of the available data sets at the time, did a meta-analysis, and what we found, I'll call your attention to this blue line here. You can see in some cases there's kind of a break right around where that blue line exists. Okay? And it actually falls out on this figure better. You can see all of the dots are kind of right along this line here. Then there's this sharp break, and then it kind of mellows out again. And this break here at about 60 meters is consistent worldwide. Okay, these data include the Red Sea, Australia, Caribbean, and everywhere we've done it, at least for, and let me back up and say this is for the benthic species. Fish are this other line, and they don't fall out quite the same way. So, but for the benthic species, the sponges and such, there is this break. And so we now have an upper mesophotic from 30 to 60 meters. This is a transitional community, okay? This includes shallow reef species that are moving down to their lower limit, and it includes the deep reef species that are moving up to their upper limit. So there's a lot of biodiversity in that transitional community, but there's not a lot of biomass because the individuals that are there aren't doing very well. As I said, they're at their limits. They're kind of, this isn't really where we want to be. In contrast, when you get to the lower mesophotic from 61 to 150 plus meters, now you're looking at a community that's very endemic, okay? Again, about 50% of those are endemic to the system. They are unique species we've never seen on the shallow reefs. We do get some depth generalists that go down this gradient, and we'll talk about that in a few minutes. But again, and then a lot more biomass, and that's because the individuals that are down there do very well there. They like it there, and hopefully by the end of this talk you'll understand why. Okay, so just to reiterate that, the deeper you go, the more sponges you get. Okay, this is a meta-analysis we did of some of our work, some of other people's work, mostly in the Caribbean, but you can see Chuk and Palau in the Indo-Pacific as well. So a pretty good phenomena here, or that sponge density is increasing with increasing depth. Alongside that, sponge volume, at least in the Caribbean, increases the deeper you go. And again, you saw that massive Zestispongia that Michael was beside in the first slide. What's interesting is in the Pacific, it goes down. And the reason is most of the sponges in the Pacific we see are sort of those little encrusting, flattened mats. They're not really these big emergent structures that we see in the Caribbean. Okay, so what about this issue of connectivity on coral reefs? Okay, you have two types of connectivity. You have horizontal, and in a study we did, we looked at, this is Little Cayman, this is Lee Stocking Island in the Bahamas near the Exumas, and this is San Salvador Island. And horizontal connectivity is just kind of riding the ocean currents from one site to another. It doesn't surprise me that Little Cayman is not related in any way to these other, that's thousands of kilometers to get there. But we're looking at about 100, 150 kilometers between these two sites, and there's still no horizontal connectivity. And this is a case of Montastria cavernosa, which is one of the more important corals in the Caribbean basin. Okay, so not a lot of horizontal connectivity, but the potential. Let's look at vertical connectivity, because this is the one that's more important for the deep reef refuge hypothesis, right? And what you can see, again, is the different depths fall out is basically entirely different. So this is different than this, than this. If we had good vertical connectivity, we'd see sort of a leapfrog. We'd see better overlap in the genomes and such, and that just doesn't come out. So from our data, we sort of came away saying, well, this deep reef refuge hypothesis may not be a good deal. The deep reefs may not, they don't have the capacity to reseed the shallow reefs, and that's a bummer. Deep reefs are still cool. There's a lot going on down there, but this isn't going to work for us. There have been a number of other studies by other genetic labs around the country and the world that have seen similar patterns to this, but there is at least one lab that I know of that works with Montasseria cavernosa sort of up here in the Gulf of Mexico, and they have seen some degree of vertical connectivity. So I don't think it's, when we wrote the paper, it was very much, well, it doesn't work. But now I'm more of the opinion that it's not a yes or no answer. There's a gray area in between, and it's probably a site-specific thing. Some places there may be some degree of vertical connectivity for probably physical reasons that we haven't thought about yet. In other cases, it just isn't working, and that's kind of where I'm going to leave that right now. But we do have to take into account this issue of depth generalists, because, again, it doesn't make any sense to be vertically connected if you're a depth specialist. If you get blown up onto the shallow reef, you're just going to die, and that doesn't help the reef at all. So this is something we want to look at. And it turns out there are some depth generalists. In the corals, Montasseria, the one I just showed you, is a good example. You can see that we've collected it down as, in fact, we've collected it down to 100 meters. But in this particular figure, you can see it falls off as you go deeper, and that's not surprising. 90% of the energy for a coral is coming from its photosymbionts, right? So the deeper you go, the less energy it's going to get. That's not a place it wants to be. It can be there, but it doesn't want to be there. The interesting thing here is you can see these spots. The percent cover is actually going up the deeper you go, and the reason is they start out as mounding corals on the shallow reefs, and they end up as plating corals like this one is on the deep reefs. They're trying to maximize the area for a capture of light. So there are some adaptations that these guys get. But even the depth generalists have some issues, and here are the photosymbionts, and you can see that they are very depth specific. So there's some issues with all of this anyway. Possibly the larvae can move up and then trade out their zooks, but I don't know. All right, I'm not a coral person. I do study sponges, though, and sponges are great because they are more depth generalists than corals, okay? This particular paper, we collected sponges all the way down to 76 meters. Again, this is a guy that I've collected as deep as 100. We've collected it back in caves. The thing about it is it doesn't have any photosymbionts at all. So it's not worried about the light. So it just grows wherever it grows. And that's the case for a lot of sponges. So they represent a pretty good source of potential propagules, if you're interested in sponge reefs, which I am. All right, what about this issue of stability? Well, we had an interesting sort of a natural experiment about 20 years ago in the Caribbean, and that is when the lionfish came in and invaded. So we had this invasion event. And prior to that, this is a place called Pulley Ridge, which is a drowned island that's about, well, it's the westernmost end of the Florida Keys. And it's a big island, so it has a north and a middle that we looked at. But prior to the lionfish invasion, these pictures we got from the USGS, you can see, there is a little bit of algae, but there's also sponges and corals and other things going on here. So we have some algae, we have 10% coral, that's pretty good. We have 2.5%, 3 sponges, so there's good numbers, what we'd expect. And then the lionfish came in, and all of a sudden we saw the algae shoot up, and that's outcompeting now the corals and the sponges, so they go down. And the same thing happens at the other site. In fact, we had seen something similar in the Bahamas a few years earlier. This is a sponge-dominated community prior to the lionfish arriving, at which point it becomes an algal-dominated community. And that's really because of this change in the fish populations. This is prior to the lionfish. This is afterwards. It's significantly different. And basically what happened is the lionfish, well, they ate everything, but in this particular case, they're taking out a lot of the herbivores. And in taking out the herbivores, the algae comes back and again, outcompetes my sponges. So in this case, stability is not so great. And I do want to say, I'm not going to show you a bunch of more data, but we have in fact seen coral diseases down on these deep reefs. We've seen bleaching on deep reefs. We've seen fishing gear wrapped around the deep reefs. We've seen pollutants that end up down there. And so I don't think mesophytic reefs are as stable as everybody would hope they would be. In fact, they're something we really need to take care of. They're a really incredible community that could be lost. All right, and one last thing before I jump into sponges in a major way. While sponges are the bulk of the community, they are not the most important player, the foundation species. It turns out those are gorgonians. And we discovered this. We were in the Bahamas. This is Bach Wall and BA Line. They're two reefs that are about 3 kilometers apart as the gull flies. And so they see basically the same characteristics of slope of the reef, the same amount of light exposure, so on and so forth. But when we got into the water, this reef, bunch of dead gorgonians, and basically monocultures of a few species of sponges. Whereas at Bach Wall, we had all kinds of living gorgonians, a lot of other types of sponges, and a very dense and diverse community relative to the other site. And so we started asking, well, why is that? And as a chemical ecologist, one of my thoughts was, well, these gorgs, which produce a lot of natural products chemistry, might be producing allelopathic compounds that knock out these competitors. And so we wanted to see if maybe that was right. And we ran a couple of tests. This one here, we basically took dead gorgonians and live gorgonians, placed them into plots, cleared plots for a time period, and then came back and looked at sponge recruitment. And you can see that hydrodynamics alone is not explaining the differences here. In fact, live gorgonians, we see significantly better recruitment at both depths, as well as more sponge species. And so that's pretty interesting. You have to have a live gorgonian present. But does that mean that there's allelopathy involved? So we went ahead and did some find them and grind them work with Elicella. And here's a crude extract, and here's 3 of the compounds we pulled out of it, 1, 2, and 3. And then we ran some assays, some cell lysis assays against this guy. This is Agilis clathroides, the elephant ear sponge. And this is one of the ones that we see in here. It's sort of made these dominant, communities. And so we were looking at cell lysis, and sure enough, at least 2 of these compounds break this down pretty effectively, suggesting that there is some allelopathic activity. And then just to sort of close the loop, we went ahead and looked at how these compounds are being leached into the water column, how far away the recruits are relative to that, and got a good feeling that, yes, this is being driven by chemical defenses. Okay, let's get back to sponges. So this is a reef. This balmy is maybe, 12, 15 feet tall and about 30 feet around or whatever. And there's a couple of pieces of coral here and there. There's a couple of gorgonians. But the bulk of what you're seeing here, the reds, the browns, these grays, these little structures, these are all sponges. So this is a typical Caribbean reef with its sponge load. And I'd say, and this is probably in 15 feet of water, maybe 20 feet, probably 60% sponge cover on this. Okay? So sponges they dominate Caribbean reefs. There are 10 times as many sponge species as there are coral species, but people are more interested in corals. They sort of act as condos for worms, for crustaceans, and other things, so they're a good source for infaunal species. Some of them, this particular guy here, do have phototrophic microbes, so they may play a little bit of a role in terms of the energy budget here. Sponge people now talk about high and low microbial abundance, and that because the bacteria are really important. High is like 10 to the 8th, 10 to the 10th cells per gram, and low is 10 to the 5th, 10 to the 6th. We'll talk more about this issue in a few minutes. And then they have a lot of functional traits. Besides being food and habitat, they stabilize the substrate. They kind of act like a glue to hold the reef together. Some of them are capable of bioeroding. Sponges are well known for their water filtration, which leads to their ability to benthic pelagic coupling. They're part of the carbon cycle, part of the nitrogen cycle, and they're huge competitors with corals and other things. So be impressed with sponges. And I just want to mention that this HMA, LMA issue is also a functional trait because sponges are sort of archaic enough that they can't do a lot of their own metabolism. So the bacteria are heavily involved as a true symbiont in taking care of some of the things that sponges need taken care of. All right, so everybody learned that functional ecology is what we need to talk about now. Okay, back in the day, managers decided we no longer want each one of you to have your own pet project. You have to deal with the organisms that are most important for patterns and processes on communities, and that's fine. I get that. And what's important then are the functional traits that these guys are having. But it's important to note that while overall they may have a positive role in the community, some functional traits also have negative impacts. Okay? And so here's our sponge functional traits. And we're not going to talk about all of these today, by the way. We're just going to talk about one. But I want to use nitrogen as an example. This was my paper that was dealing with cave sponges. I showed you I was interested in cave sponges for a while. And when the sponges pee, that nitrogen ended up out on the reef, and the corals were taking it up, and they were growing better, and they were healthier, and everything was good, so nitrogen good, right? But this is one of those double-edged swords things because nitrogen is also a fertilizer for algae, and we were talking earlier about the whole phase shift issue, so nitrogen bad. So again, these functional traits are kind of a trade-off that you got to deal with. Good news is I'm not dealing with nitrogen today. I am actually going to focus on benthic pelagic coupling and to some extent water filtration of the sponges. And that's because it has huge implications for nutrition with the detritivores and the predators. But again, while there's a positive, there's some competition issues involved as well, and we'll talk about those going forward. Okay, the currency that we're going to talk about with sponges relative to energy is DOM and POM. Okay? And depending on where you are on a reef, shallow reefs have a lot more DOM and a lot less POM, and it's the exact opposite the deeper you go. This is not a problem for sponges. Sponges can take up both across the entire gradient. It's just what they have available at any given point. Okay? And what is important is DOM is heavily involved in this new theory called the sponge loop. It's not really a theory anymore, and it's been around for over a decade. But Jasper's group showed that corals and algae are releasing DOM. Sponges take those up. Sponges grow. As they grow, they shed off old cells. That becomes POM that is available to the detritivore community. So there's a throughway of energy that involves sponges that's really important on these reefs. Bear in mind that we've always known sponges are, the tissue is being eaten by turtles and angelfishes, so predation. So there's energy going into those groups as well. And as I said, there's this issue of competition because sponges filter a lot of water. And they, on their own, they're not just looking for DOM, they're taking up POM. Okay? And the POM they're taking up may be POM that the detritivores want. And so one of the things I'd like to get at, we haven't figured it out just yet, but the POM that they're giving them back to the system, is that more or less than the POM they're stealing from the system? That's an interesting question that we need ultimately to totally understand the energetics. But you can see this is a very complex system, and sponges are dead center of it, and that's pretty interesting. Okay, just to reiterate, sponges are pumping a lot of water. Okay, here's 3 of our lab rats, Agilus, Placortis, and Zestospongia. They're all HMA sponges. Doesn't matter, we've done this with LMA sponges as well. And what you can see here, very simply, is the bigger the sponge, the more water it pumps. That's somewhat intuitive. Why are you pumping water? Well, it's the trophic resources within it, right? And so, again, DOM, it's dropping as you go down the reef. POM is increasing as you go up and down the reef. There are other cellular constituents that the sponges are capable of picking up at the same time. By the way, this is data from a study we did in Curacao, but this pattern is something that is totally reproducible within the Caribbean. Okay? I can show you data from Belize, Bahamas, Caymans, Jamaica, all showing very similar patterns. And so this is what the sponges in the Caribbean basin are dealing with. Okay, this is their reality. What about their metabolic capacities? Well, we did a study and we looked at HMA and LMA sponges, 5 of them from 4 sites: Belize, Curacao, Caymans, and St. Croix, and threw it all together with their metabolic capacities. And it's interesting how it falls out, because these groups are LMA and this group is HMA. And HMA sponges they have a thicker tissue, if you will, so they're not as capable of filtering as much water as the LMA sponges. So they're going to basically focus on DOM. That's what they prefer. Again, they're able to take up either, but by and large, HMA sponges are the ones that are going to do the DOM. From the POM standpoint, LMA sponges have less dense tissue, they filter more water, and they're able to take in the bigger particles, so they're going to focus primarily on the POM. Okay, and then they grow, right? You take in food, you do something with it. One of the things sponges do is they grow. And so here is, again, Agilis in the Caymans, and I don't know, there's 30, 35 data points here. We tagged these guys and followed them for a year, and so you're looking at growth rate, and the deeper you go, the faster your growth rate. Okay? And that suggests that something on the mesophytic reef is better. Remember, there's more POM down there, even though these are HMA sponges, they're still able to take care of the POM. There's probably more energy down there. They like that. In the same paper, we went ahead and did some reciprocal transplants, 22 and 61 meters for 18 months. This is just one of the tubes here. This little white marker is for us to look at growth over time against that. And what you can see is, if you're on a deep reef, you're growing you have positive growth over that time period. This is deep to deep, and this is shallow to deep. So the deep reef sponges are growing well. The shallow sponges, no growth or negative growth over that same time period. So there's a difference there in terms of energy and food availability. And then we've also looked at, using stable isotopes, how these sponges fit into a broader community. I'm not going to go into this too much, but these green squares are Agilis. And you can see they live in a different trophic niche than these blue triangles, which are the big Zestaspongium things. So sponges, even though they all sort of do the same thing, they all sort of filter water, they're taking up different constituents, and they're doing different things with them, and it's a very interesting mess. Okay, once they have that energy and they've grown, now they're available to produce food, right? They're going to give food to detritivores, and they're going to give food to fishes. Okay? The detritivores, again, that's part of the sponge loop. They release detritus. There is a component called phytodetritus I haven't talked about yet, but the important thing here is you can see when you're on the deep reefs, they're releasing a lot of phytodetritus. If you're on the shallow reefs, not so much. Right? So again, if you're a detritivore, a good place to be is also on these mesophytic reefs. Fishes are eating the tissue itself. We have some generalists, the butterfly fish, a file fish, and a puffer fish. And then we have 3 specialists. These are angelfishes that feed primarily on. And what you can see is the percent of bites on sponges, shallow versus deep, they're pretty consistent, shallow to deep. So fish bite about the same amount. The difference is on the deep reefs, they're going after more sponges, 25 versus 35, 10 versus 38. And that may be a factor that deep reef sponges tend to have less in the way of chemical defenses, not something I'm going to get into today. It might be because there's more biodiversity down there. There's just more options for them to taste. I can't answer that right now, but it is pretty fascinating, I think. All which brings me to sort of the end point here, and it's, I've for years, I've seen data come out and say, well, on my little reef that's on this corner of this island, that's on this archipelago in this sea, things look good or things look bad. And I wanted to get to a point where we could say, are the sponges doing anything that globally are important? And so what I did was I wanted to scale up to the level of Grand Cayman. And if you take the circumference of the island, that's actually about 120 kilometers. And the sponge belt, again, 60 to 90 meters, that's going to be the lower mesophytic. We have really good data for that. Turns out at about the middle of it, it's about 70% sponge cover, about 4 sponges per meter squared, and their volume is averaging about 8,000 cubic centimeters. Scale those numbers up, and it says that there are 13.5 million sponges in that sponge belt. And they represent almost 300 million cubic meters of sponge tissue. And they're able to filter effectively about 1.5 million liters of seawater per minute, from which they're removing 0.7 of a mole of POC, 1/10 of a mole of PON, 100 moles of DOC, and 3 moles of DON per minute. Obviously, once they've got that energy, they're producing detritus, about 1.3 grams per meter squared per minute, and phytodetritus, about 1 gram per meter squared per minute. So I come from a biochemistry background as well, and I wanted to torture my student, so I made her do most of the work. But you can turn that into energetics, right? And so the energy of the standing stock of these sponges here represent 8.1 times 10 to the 11th kilojoules of energy for the predatory angelfish and turtles. And then the detritus and phytodetritus, 10.5 times 10 to the 7th, 7.8 times 10 to the 7th kilojoules per minute for these detritivores. And I went, wow, that's big, right? There's a lot of zeros after that, and that's about as far as I can get. I don't know what that means. So I turned it into something I could have, that's pepperoni pizzas, right? So it turns out each pepperoni pizza is about 11.75 kilojoules. And if you do the math, it turns out this standing stock of sponges represents about 69 billion pepperoni pizzas sitting down there for turtles and sponges, okay? The detritus and phytodetritus together represent another 15 million pepperoni pizzas being produced every minute. So that all of a sudden starts to sound like a lot of energy that's available to predators and detritivores. And that, bear in mind, this is just in the sponge belt from 60 to 90. In fact, the sponge belt in Grand Cayman goes down as deep as 120. We haven't done a lot of work down there. So, but, you know, we're probably underestimating there. And then you think of the fact that this is just in Grand Cayman. What happens on Little Cayman and Cayman Brac? We're in the midst of writing up that paper right now. So there's a lot more that's going on there. And what about if we take it beyond the lower mesophytic and include the upper mesophytic and the shallow reefs? Sponges are a ton of energy, right? Just in the Cayman Islands. And anybody who's ever looked at a map of the Caribbean knows that Cayman Islands are literally a speck. They're dwarfed by Cuba right above them, and Jamaica kind of makes the grade just kind of below them. But you then think about all of that coastline around the islands, the Greater Antilles, the Lesser Antilles, and then sort of the shoreline, and you start to say sponges are hugely important, right? So in conclusion, as I said up front, I don't think that coral reefs are going to become sponge reefs, but it's clear to me that the functional ecology of coral reefs is largely due to the sponges and their microbes. One of the more important aspects of the sponges is they bring in the benthic-pelagic coupling, which allows them to take DOM and POM that would otherwise probably be lost and turn it into a source of nutrients for detritivores and predators. And then if you do back-of-the-envelope calculations like I did here, you can see that the significance for the Caribbean and potentially for other ocean basins as well is pretty fantastic. And with that, there were a lot of students, postdocs, and colleagues who've helped on various aspects of these projects over the years. We got funding by NSF and NOAA. Gordon Conference funded us to talk about it. Various marine labs have put us up, and thank you for your attention.
Okay, thank you so much. Do we have questions from the audience?
Math is overwhelming.
That was really interesting, Mark. I was looking at your energetic calculations and thought, what if it's the standing stock? Because that energy, I mean, that was impressive just at that band.
Yeah.
But they also live longer, I mean, compared to algal turnover. And then, I mean, I guess corals would be is
Right.
more stony, so there's not as much organic material.
So I didn't get into it here, but I showed you that slide with Placourtis, and we were able to see the depth gradient of Placourtis. That was actually a study that was actually looking at tissue regeneration. And so, yeah, you're right. A standing stock could disappear through time, and the turnover isn't like it is in, but you actually get tissue regeneration. And it turns out that because of chemical defenses and other things that a lot of times something takes a bite of it and then they leave it alone because now it doesn't taste so good and it gives it a chance to recover.
Oh, I was actually thinking that it was more prolonged, that it was more long-lived. So it's interesting.
Hi, thank you for the talk. That was awesome. So a question for you regarding the the stability of, I know you talked about these mesophytic, these reefs that were primarily coral dominated. What do you think could potentially be a sort of like level of resilience when it comes to a potential sponge dominated reef? Like, do you think it's more or less likely to be resilient to another phase shift?
So, in my experience, you have coral bleaching or diseases that attack the corals. Sponges are doing pretty well. We've never seen a situation where all of a sudden once that space is free, Diana's algae is what moves in because they grow really fast. Sponges are a little slower, but the sponges aren't getting taken out by the stressors as much. So in the long term, I think there's a chance that, yes, we will see, some degree of sponge reefs. But I don't think there's going to be this massive overnight shift where phase shift where you go from, 20, 30% coral cover to all of a sudden 20, 30% sponge cover. I think there'll be winners and losers, and I think over time things will settle out and we'll get back to some kind of stasis that'll exist for the future.
Awesome. Thank you.
Sure.
Thanks for the talk. I was hoping you might take a trip back down memory lane and return to like your temperate mindset and your time at Moss, and whether you have any thoughts on the potential promise of a deep reef refuge. There's a lot of conservation interest for that as a potential positive thing to hope for in the context of increasingly frequent and more intense marine heat waves, for example.
Right.
So I'm thinking of kind of mobile taxa like Pycnopodia, for example, like sunflower stars. Is there any potential for refugia on along the west coast of North America, for example?
Yeah, so mobility is an interesting factor, and you saw in our analysis that fish don't, fish can swim away, right? If there's a problem, they just swim deeper, they swim to another site. That's an issue, and I think that's where your Pycnopodia are as well. It may very well be that they choose to go deeper, and that would make some sense. And we know already that there's fishing refuges for the abalone. If you get below, skin limits, most of those abs are probably okay until they see their next disease and stuff. So yeah, at some level there's a refuge, potential. I think probably in every system. It's just how it works, and potentially that is one that So for instance, with abs, you can imagine a situation they're broadcast spawners. If they're all down deep, and then maybe they reseed shallow reefs, and the next generation of ab killers come along and take them out again, sort of things.
Is there are there any active groups that you're aware of that are studying that depth band along our local coast?
Diana would probably know better. I mean, is anybody doing deep diving in? Well, just deep reefs.
There are a lot of non-science.
The Cal Academy has a
Oh.
mesophytic reef.
Yeah, but so Louis Roca is a good friend of mine. In fact, he was part of this as well. But they go to the Philippines. I mean, they go everywhere. Their trips are awesome. Ours are kind of budget, but But yeah, they go to a number of places, but tropical. He doesn't do a lot, I think, so much off of the California coast.
And actually Heather Spalding,
She was also, yeah,
she worked on the deep reefs off of Stillwater Coast, deep work.
yeah.
But, I mean, CSU and the other group is doing pushing out that. But there's a lot of room for work.
Thank you.
I have several. I have one though. I'll ask with the group. I love the pepperoni pizza analogy. So to stick with that, I don't like anchovies on my pizza. How many sponges do you think proportionally are chemically defended, like having an anchovy pizza? Like.
So we actually, there's a group based out of UNC Wilmington, and Joe Pollack has done a lot of work on, he sort of tested everything. I have some issues with some of his work, but the bottom line is he's got like 80 odd species that he's then said defended, not defended, marginally defended, or whatever else. And it works out that about 50% of the sponges in the Caribbean seem to have good chemical defenses, and there's another group that their strategy is don't invest in the chemical defenses, go ahead and grow fast kind of thing. So
That'll be neat to consider in the energy budgets of like palatability of sponges or whatever you want to call
Right,
it,
right.
which ones they want to eat.
yeah, no, that's
Yeah.
right. It'll change the amount of available pepperoni pizzas. It might be only 30 million or 30 billion sort of thing. So we'll see.
Okay.
It's
It's
still a
still-
lot of pizza.
It's a lot of pizza. Any other questions, including from online? No? We're good? Okay. Okay, one more from me.
Sure.
I was curious about two things: the LMH, the low and high microbial community contrast, and then the metabolic capacity panel that you showed. So first, are you referring to like numbers of species in when you say low versus high microbial community? I'm sorry, what's the term? LMH, HMH?
Yeah, low microbial abundance and high microbial abundance sponges. And that just has to do with how many bacteria they have per gram of tissue, essentially.
Okay, so it doesn't have anything to do with the diversity of, ASVs or whatever, or
No,
OSUs.
not so
and,
much.
Okay. And then metabolic capacity. Could you explain what that metric refers to?
So as I said, it's really the bacteria that are driving the metabolism of the sponges in terms of breaking down nutrition and other things. And so you can't just look at the sponge alone and say this is doing this. I mean, we now recognize that they're, so using just sort of keg and go analyses, you can actually go in and look at, okay, here's the whole microbiome of the sponge. What are they capable of doing? And the metabolic capacities. So we threw all of those in together, and that's how you got that one figure, that NMDS plot, where LMAs are different than HMAs. There's different metabolic capacities, but that's hundreds of metabolic functions that are occurring simultaneously. And it's different, and part of the difference may be because you're feeding on or I shouldn't say feeding, you're feeding on everything, but you're focusing on different components of the water column nutrition.
Thanks.
Okay.
Okay.
Thank you so much. I think we'll stop it here, and if you have more questions, please feel free to come up and talk to Mark about any aspects. So let's all give him a round of applause and thank him for coming.
Thank you.